Well, okay. Hello, everyone. It's connected. Yeah, I did it. It's connected. Just checking the sound. So it is my great pleasure to welcome all of you here to Civil in Vairão, near Vila do Conde and Porto, to host this event. So this event marks the delivery of genomic applications that we've developed in the Biodiversity Genomics Europe project. And in this introductory talk, we would just, Rob Waterhouse and myself, I'm José Mel Ferreira, by the way, we will just set the mood and the stage for the rest of the conference. I just would like to thank all of you for coming here in person, and I want also to thank everyone that is online watching this this conference. And so we can start. So yeah. Go ahead, Rob.
Okay, testing 1, 2, 3. Yes.
Don't forget to share the screen.
Share the screen. I let you do that. Okay, we're good. Oh no, now we have to press it. Okay, so thank you very much, José, for the kind welcome, and welcome to you all from me, Rob Waterhouse. As José said, we have this little opening session just to set the scene. You, in the majority in this room, are very well informed about what is ERGA and what is BGE, or you should be by now, I would say. So bear with me for the next 10 minutes, because we do have a lot of viewers online who are maybe less familiar with what is ERGA, the European Reference Genome Atlas, and what is BGE, the Biodiversity Genomics Europe project. So I am, let's say, mostly speaking in the next 10 minutes to the people outside of this room to, again, set the scene of why we are all here today and for the next couple of days. And then José will then dive more deeply into the application side of things, which is, the main purpose of us here today. So the conference is organized by ERGA and BGE. ERGA, the European Reference Genome Atlas community of scientists, and BGE, the Biodiversity Genomics Europe Horizon Europe funded project. So ERGA is a community of scientists trying to work together to coordinate the sequencing and assembly and production of what we call reference genomes for European biodiversity. And as such, we represent Europe, so we are the European regional node of the Earth Biogenome Project, which is the global umbrella organization coordinating the production of reference genomes for the entirety of eukaryotic biodiversity. And as we will see in the next couple of days, ERGA, we are not just about motivating and coordinating the production of reference genomes, but we are increasingly thinking about also how we foster the use of these reference resources to answer important evolutionary, biological, bioeconomic, and conservation questions. And so Biodiversity Europe, Genomics Europe, ERGA came together with the barcoding community, so here represented by IVOL Europe, to write a proposal for a grant, which is now in its 3rd year of running, bringing together multiple institutions from across Europe and the barcoding community and the reference genomes community together in this common project. So we have the ERGA community and we have the BGE project. So the ERGA community is made up of more than 1,000 individual researcher members from across Europe in this kind of bottom-up initiative, organized with a governance structure, with a council representing all of our 38 member states, an executive board who tries to steer our direction, and the committees, which is where really kind of the day-to-day work happens in our community organization. And we're kind of, let's say, guided by three overarching pillars of activities. The first being, as an initiative, we are focused on the generation of high-quality reference genome resources for biodiversity. At the same time, we act as the umbrella for Europe. So there are lots of different initiatives going on, some big projects, some of which you may have heard of, like the Darwin Tree of Life in the UK, or the Atlasy of marine species in France, so two of our biggest projects, but all the way down to individual researchers, individual research labs who are also contributing to the effort of producing this catalog, this atlas of reference genomes. And thirdly, we act as a kind of knowledge exchange hub. So we really try to create the space, the forum for researchers to exchange what's working and what's not working in terms of DNA extractions, in terms of sequencing, assembly, annotation, all the different steps you need to achieve your reference genome so that we can work together to get there faster, basically. And so just in terms of the governance, we are extremely inclusive, and we want representation from across European countries, with 38 member states represented on the ERGA Council. And this for us is really important because we recognize that across different European states, there is a very different level of maturity in terms of the applications of genomics technologies, also in capacity, in the infrastructures that are available. And so we really want to bring everyone together around the table to help those that are still struggling to get started by learning from those countries that are already quite advanced. So that's really a guiding motto, if you like, behind the ERGA community. In terms of the different steps involved in producing a reference genome, the first thing you think of is the genome assembly step. But there's a lot that comes before, and there's even more that comes afterwards. So we're really trying to coordinate efforts all the way from the sampling, the taxonomic identification, the DNA, the RNA extractions, through to the sequencing of the long-read data, the scaffolding data, then building the assembly, then using the RNA-seq to annotate those assemblies and stick them into the public open repositories, with just a couple examples of our community sampled species there on the right-hand side. And where the kind of body of the work happens in ERGA is through our committees. And so these are, I see many of you in the audience are active members of some of our committees. This is where the work happens. So it's getting together and talking about standards, talking about building guidelines to help others achieve what it is that we are aiming for. Sharing information about what's working, what's not working, trying to build in support systems so that you can ask your question and someone else out there in the community maybe has already solved this problem and can provide you with a quick answer so that you can get moving much faster. So yeah, building these protocols, building these best practices, standard operating procedures, and sharing them across the community so that everyone can achieve their reference genomes. With the kind of top row of committees there focused on the production side of things, from sampling, sequencing, annotation to data analysis, but also supported transversely by, for example, the ELSI committee, ethical, legal, and social issues, our citizen science committee, which is, perhaps should be named citizen science and stakeholder committee. So it's really much broader than just citizen science. So how can we also, through the media committee, communicate the importance of genomics and the applications of genomics in a wide array of situations. So now to move on to the BGE project, Biodiversity Genomics Europe. Here, just a reminder, we're coming together with the barcoding community with three overarching goals. So building capacity across Europe for biodiversity genomics, increasing production in barcode catalogs and reference genome catalogs, and the third pillar, which we will be talking about a lot in the next 3 days, is demonstrating the use of these new genomic data for applications in biomonitoring programs. So we have the barcoding stream and we have the genome stream in BGE project. And perhaps at first glance, you think these are kind of two different technologies doing quite different things. But when you think about it, we do have a similar workflow or a similar value chain, right? We have to do the sampling, bring the samples into the lab. We have to do the sequencing, then the data to applications pillar, and finally the applications, which we are going to be talking about the genome, the reference genome applications the next 3 days. And here, with this last or next to last slide, it's really just to talk about the interaction between a European project on the one side, so BGE, and a community or ERGA on the other side, where the activities of the project are helping to fuel the advances in the community, but also the needs and questions coming out of the community are helping to shape the priorities of what is being tackled within the European project. And so, you know, there's a nice symmetry there, and most of you here in this room are effectively part of both sides of this coin, the project and the community. And so with that, I would like to thank you all, especially for being here today. I thank everyone who's joined online to learn about biodiversity genomics and reference genomes. It's a really fantastic community to be part of, and I really look forward to hearing the results and the progress of all of our different case studies across the BGE project. And I guess I can hand over now to José for that more application side of the story. Thank you very much.
Thank you, Rob. So the next couple of slides will sort of set the mood to why are we all gathered here today in the next couple of days. Most of the things that I will say will not be novel to anyone in the room, but I hope also it can set the stage for people online that are less familiar with the work that we're doing. And essentially, what brings us together is the importance of biodiversity. And biodiversity is important not only at a fundamental aspect in terms of sustainability of ecosystems, but it is also very key to sustain human society and a proper and sustainable use of these resources. And it sustains health, resistance to climate change, food production, and everything that we can use from nature. However, also another thing that brings us here is the fact that biodiversity is declining at alarming rates. There are diverse human activities related to ocean acidification that cause ocean acidification, climate change, land use change, diseases, habitat destruction, overhunting, and human population growth that have been inducing strong declines in biodiversity, not only at the species level, but importantly also at the intraspecific level, at the levels of diversity that we can see within species, across populations, and within populations. And estimates point that we are perhaps 1,000 times, we have extinction rates that are 1,000 times faster than the regular background extinction rate that we would expect. And so this urges the use of new technology, new scientific developments to tackle these alarming biodiversity declines that are causing not only the breakup of this ecosystem stability, but also the putting in question the sustainable use of these resources, these natural resources. And so we are here gathered to showcase and demonstrate the power of reference-based genomics to tackle this societal question that is urgent today. These are a couple of papers that came out from the ERGA community discussing why reference-based genomics can be used to tackle the biodiversity crisis and help biodiversity conservation. But essentially, reference-based genomics, meaning the use of high-throughput genomics data that can cover the whole genomes, but anchored on high-quality reference genomes for the species that are being analyzed, is this sort of strategy is very powerful because it allows us, for example, to assess and monitor genetic diversity and population structure at an unprecedented scale relative to what was previously possible with more traditional genetic tools. identify adaptive variation and evolutionary potential, meaning that we can, by identifying the pieces of the genome that are responsible for local adaptation, we can incorporate this into the population and biodiversity management. Detecting hybridization and integration, so the fact that different species can sometimes interbreed and there are pieces of the genetic diversity of one species that can cross the species boundary. So detect this, identify this, and incorporate this into the conservation of the species. Informing conservation management decision, defining very precisely conservation units, set the stage for effective translocation planning, habitat management, etc. Merging also not only the study of specific species, but also the interaction between hosts and parasites to provide a One Health perspective to the management of the ecosystems and of biodiversity. And of course, anchoring sustainable use in bioeconomy by developing tools that allows us to have a sustainable use of the biodiversity resources that we can use in our society. And then there are three dots in the end, because the potential of reference-based genomics is immense, and these are just a few examples. Despite this potential and the growth in the scientific developments that led us here now to the possibility of using these new technologies, incorporating genomics into practice is still very challenging, and that is also part of the objectives that brought ERGA and then brought BGE into place. And here in this graph, I'm showing you just a snapshot of a gap analysis that we've developed in the BGE project. That here, the numbers that are there are based on 1,000 papers that claim to have developed a genetic application. And you can see in the green line that these are low throughput genetic applications, so meaning using a few markers, traditional genetic techniques. And this, of course, have been growing since the end of the last century. And then high throughput approaches that are here, the line in red, that have started to be applied much later, but are quickly catching up with the low throughput approaches. Yet this is a scientific development that still lacks the translation into a proper translation into practice. And in this gap analysis, and I think we all as specialists recognize that there are still many challenges to take the scientific developments in genomics into practice. One, and that Rob mentioned also briefly in his presentation, is capacity building and mitigation of cross-country inequalities. We saw in this gap analysis that the use of genomics is very unbalanced across countries, and so this is a major challenge. Importantly, scaling reference genome resources, because another thing that we saw is just only a subset of these genomic applications are actually using high-quality reference genomes from the species that they are studying. An important challenge is also harmonizing and standardizing genomic analysis pipelines. There are many, and everyone has their favorite way of analyzing the data, but to translate this into applications that can be widely used, even by non-specialists, we need to standardize and harmonize these analytical approaches. Then we have also improved the aspects of the transferring this knowledge and effectively engage stakeholders, not only as a receiver of our results, but in an engagement that starts early on in setting up what are the relevant problems to be addressed. And then finally, of course, incorporating genomic indicators into policy frameworks. This is yet a road that we need to follow to communicate with, to have this transition in scientists, stakeholders, and policymakers to make understandable how genomics can be used to tackle all these problems that we are facing today. And so some of these challenges are or all of these challenges have been the motivation for the development of ERGA, and in particular by BGE as well, and then within BGE of the development of the genome applications. And here I show you very briefly the structure of the work package that dealt with genome applications. And so the idea was to showcase how genomics can actually be used to tackle relevant practical questions in conjunction with stakeholders. And so this work package was structured in sort of two types of tasks: the applied case studies that do the scientific research and development work that incorporate reference genomes and associated genomic data to answer to very specific questions that are relevant to a certain species or a certain biodiversity problem. And then two other tasks that support the development of the applications. One, the developing networking and other activities that maximize the applicability and the communication also among the researchers that develop the case studies. And another task devoted to establish the link and support the establishment of the link with stakeholders and public engagement. And so these are shown in the bottom within the case studies, and we will see during these days the results of most of these case studies that were developed. These were divided into two main topics: those that are devoted to conservation genomics, and others that have a topic more related with bioeconomy and disease, but all have in common the fact that they are producing applications that can help us manage and promote the sustainability of biodiversity in the specific cases. And so during today and tomorrow, and then on Friday, we will have roundtables to discuss many of these challenges that I presented before. We will have we have built in BGE a portfolio of genome applications. Here you see many of the pictures of the model species that were used in each of the case studies. And I'm very much looking forward to hear about all the results that all the teams that were involved in the different case studies have produced. So this is a very brief overview of the program. Note that the time is Central European time. So for those being here in person, just remove 1 hour from this program. And I know that this is sometimes confusing, but that's this program for people that are online is in CET. The local program that you all have, the link in your badges, is in local time, okay? And so today we will have, so in addition to the case study presentations, we are fortunate, and I would like also to thank our keynote speakers. We will have during the conference 4 keynote addresses, and we will have sessions devoted to presenting the results of the case studies that, I remind you, not only involve the scientific developments, but also the activities of stakeholder engagement that were done during the development of the case studies. And then in the last day, we will have roundtables and also a series of parallel sessions to close this conference with flash talks, people submitting their talks. And I would like also to thank the community in general for having been so enthusiastic in submitting these talks that we will have at the end. One important aspect is the code of conduct. Here we will adopt the code of conduct that is online, and you can search from Elixir, and essentially just use common sense and respect diversity, foster trust and collaboration ethically and with integrity, and show respect and courtesy to each other. I think that's the normal way of working in society, and I hope and I'm sure that we will all commit to these principles here during the conference. For people that are here, so here as you can see in this ground level, we are just very close to the outside. So if there is any emergency, just leave. But to there. But when you are, and especially in the last day where there will be some sessions in rooms upstairs, please take note of the fire escape routes. And there is this emergency plans that are placed in several places of the building upstairs. So, and be aware that when you go there, before you enter the rooms, just check your escape route in case something happened. But of course, nothing will happen. Everything will go well. So I have nothing left to say, just to wish you a great conference. I'm really happy to host you all here. And I would like also to take this opportunity to thank the people that organized this conference. João Pimenta as the main local organizer. I mean, I'm here speaking, but he organized the meeting. So thank you very much, João, for organizing this wonderful conference. Also the team from BGE, Christian, Luisa, Chiara, that were instrumental in organizing this. Also people here locally from my group that have devoted part of their time during the last weeks also to help making this conference and the organization a success. Thank you all for coming, and I'm looking forward to the rest of the conference. Thank you. Yes, I think so. Yes.
Is it on? Okay. So, it's already there. Very nice. Yeah. Okay. Well, first of all, I wanted to actually say hi to everyone and welcome. And just a very brief words. I'm Camila Mazzoni, for maybe those who don't know. When we were thinking, Claudio, Rob, and I, in the very beginning, how to build this project, together with our colleagues from IBOL, the applications part was something that, was not the major or the central part of the project, but it was something that we really insisted to be there because we saw that this was extremely important at that time. And it really paid off because now we know how important it is. And we are really happy that we have so many case studies, so many projects that are associated with this project and that are going to show the world, and in this case, Europe, hopefully, how genomics can make a difference. So thank you very much all for the trust on us, for joining the project. This has been really a great surprise that this worked out. Thanks a lot, José, for leading the work package 11. I know it has been also a challenge, with dealing with so many different people, and of course Robert and Christian with the extra case studies that we got along the way. So yeah, this I'm looking forward so much, very looking forward for what we're going to hear today. And I'm really honored to introduce you to our first keynote speaker, Christina Hudson, who is going to represent also Gernot Segelbach here. And I would like to tell you a little bit about Christina before she joins us here to give her talk. So Christina is passionate about uniting science, management, and policy. She coordinates the EU Biodiversity Plus Genome project, working to integrate genetic indicators into EU and global biodiversity frameworks of the UN Convention of Biological Diversity, CBD, and actively contributes to initiatives like the EU Cost Action Networks, GENOA and GBIKE, Coalition for Conservation Genetics, and the IUCN CCS Conservation Genetics Specialist Group, ensuring genetic diversity as a voice in global conservation efforts. Representing the IUCN Conservation Genetics Specialist Group, she will be presenting on integrating genetics into global policy and commitments. Thank you so much, Christina, for accepting our invitation, and the floor is yours.
Thank you for the nice introduction. Good. I'll just try and keep track of time here. So while coming here, I was kind of reflecting on what the purpose of this conference is and the plethora of nice case studies. I consider myself as a researcher as well. And what we often do is that we have a focus on a species or set of species. We collect data, we analyze it, and we have interesting results that we then go on and publish in scientific papers. Some of us even reflect on these results and, well, think they might have implications for conservation. So I would like to start with a kind of a little survey here. So those of you who have had papers, studies, where you've argued that your findings hold implications for the conservation of the species or set of species that you're interested in, and written that in the scientific papers, can you please raise your hand? Oh, that's very encouraging to see. Okay, then we take it a step further. So how many of you have then actively taken those results that you've generated that have implications for conservation and worked together with stakeholders, with practitioners to actually implement your results in the conservation efforts of the species, whether it's management or strategies, action plans for the species? How many of you have actually done that? Can you raise your hand? Okay. Well, okay, so a little less. Fair enough. Fair enough. So we would call that kind of a bottom-up approach. And what we do as researchers are that we work on the bottom, and that is wonderful. And there's a lot of great research being done. But not a lot of that research and the important results that you have that have implications for conservation actually makes it towards the top. So what I'm going to tell you about today is the opposite way around. So instead of a bottom-up approach, we're talking about a top-down approach. So how can we actually make sure that genetic diversity is considered in global policy and frameworks where there is an incentive and a requirement for countries to actually monitor and assess genetic diversity and use it actively to safeguard biodiversity? I think there's no need to say that there are 3 pillars of biodiversity, and one of them is genetic diversity. And you all know that genetic diversity is important for fitness, for resilience, for evolutionary potential of species and populations. Fortunately, that has also been recognized in the multi-treaty of the United Nations, so the Convention of Biological Diversity. And I'll come back to that in a second. What I'm also going to talk about today is the International Union for Conservation of Nature and what is done in terms of genetics and incorporating of that into, for instance, assessments. So the Convention of Biological Diversity came to existence in '92. It's a multi-treaty agreement where there is 196 signing parties. So meaning there's 195 countries, and then there's the EU. So what goes into the CBD, Global Biodiversity Framework, really holds implications for countries and their incentive to actually do something about genetic diversity. The CBD, Global Biodiversity Framework, works on a 10-year cycle, basically, where based on the new Global Biodiversity Framework, you have kind of 10 years, you have to do a national biodiversity strategy and action plan, and then there are reporting cycles where you need to report on whatever is in the Global Biodiversity Framework, and of course aligned to your national biodiversity strategy and action plan. But genetic diversity have had a bit of a rough start in the CBD. Prior to 2020, it was written into the CBD, but it was predominantly focused on domesticated species, wild crop relatives, economically important species. Some of you might have heard the AHG targets. So genetic diversity was in the AHG targets 13. And in the reporting based on the past Global Biodiversity Framework, countries were also to report on genetic diversity that was in the AHG target 13. But there was not really a fantastic outcome of that. And collectively for EU, there was really no progress in terms of genetic diversity. Also looking at the reportings based on the past Global Biodiversity Framework, the countries kind of struggled a bit, to say very diplomatic, in terms of reporting for the different sets of species. There was a major gap in the previous Global Biodiversity Framework, and that was that wild species were not represented. So genetic diversity of wild species was not mentioned. So a group of scientists and others globally came together and decided to put efforts into really pledging for genetic diversity of wild species to be incorporated into the CBD. So there was a lot of work, which I won't go into detail with, but there was a lot of scientific papers, policy briefs, interactions with policymakers, webinars, workshops, you name it. A lot of work went into it. Unfortunately, the outcome was that genetic diversity is now, for the first time for wild species, incorporated into the global biodiversity framework of the CBD. So this is super important because then the countries need to report on genetic diversity of wild species. One thing is that it says so in the text, but there needs to be a way for countries to actually assess genetic diversity. So indicators for genetic diversity was suggested, and 3 indicators were suggested. Only 2 made the cut. So it was the effective population size of 500 that made the cut, and then the number of populations maintained. But only one of them, indicator 1, so effective population size 500, is a so-called mandatory indicator. So it's a headline indicator. The other one, the countries can choose whether to report one or not. The strength or weakness, depending on who looks at it, of these indicators is that they can both be assessed using non-DNA-based data, so-called proxy data, or DNA-based data. Indicator 3 that did not make the cut was the number of species and populations in which genetic diversity is being monitored using DNA-based methods. So unfortunately, that didn't make it back in 2022, but there is still the opportunity to add indicators. So yeah, there is still hope in there. Good. So what has happened since 2022 is that the countries have, or some of the countries, not all, actually we're up to 60 now, countries have actually submitted their national biodiversity strategy and action plans. And based on that, they need to do reporting. The first set of reportings are due in February next year, and the other one in 2029. So the countries need to report on the genetic indicators of the CBD. So being involved in different initiatives in Europe, focusing on enabling countries to actually report on genetic diversity indicators is kind of a 50-50. There are some countries that will definitely be able to report, but there are also others that will make a pass and hope that they will be ready in 2029 to report on that. So I've been telling you about these genetic indicators using both proxy-based and DNA-based data, which is good for certain things. So it is a quick assessment methodology, and it can give you the genetic status of populations and species. But you will not get the finer details. And that is a message that we're actively conveying quite strongly to the countries. So if you want to really inform conservation management, you need so DNA-based studies. So in terms of inbreeding, genetic load, hybridization, but also genetic monitoring over time. So this is just one of these examples on papers where this has been stressed. Another paper that also just came out is about an approach for integrating molecular tools into biodiversity conservation. This is a figure that kind of illustrates what type of data do you need in terms of answering specific conservation-related questions. And that is the paper by Laura Patola and colleagues. Genetic diversity is not just on the kind of the multi-treaty international policy level. It also needs to trickle down. A huge focus has been put forward by the EU on restoration. And fortunately, the EU nature restoration law was passed. So now it's an EU nature restoration regulation. that has come into play and that countries, the EU countries, need to adhere to. We had a look at the draft text, and unfortunately, genetic diversity was not well represented in this new EU nature restoration regulation. But we put out a paper pledging for and also suggesting where the text could be improved to be inclusive in terms of genetic diversity. We also are part of the former EU Cost Action Network, GBIC. We had a look at the genetic monitoring ongoing in Europe. So we basically compiled everything that was out there in terms of both DNA-based and non-DNA-based snapshots, so single-time studies, studies over a temporal scale, etc., and looked at how much genetic monitoring that was actually ongoing. And also saying that it was not only peer-reviewed papers, we also searched the grey literature. There was quite an amount of work to do. But what we found is, and we ended up focusing on kind of what we call the strict monitoring, meaning that it had to use DNA-based approaches, and it had to be on a temporal scale, so at least 2 time points. And what we saw that there was not really that much genetic monitoring ongoing in Europe. And the cases that we did found was kind of the usual suspects. So it was the large carnivores, it was on charismatic species like birds, etc. So the situation did not look, let's say there's room for improvement. Yeah. So we all know, and that was also mentioned just before, that we're losing genetic diversity at an alarming rate. Also, as part of the GBIC Cost Action Network, we did a meta-analysis. So we pulled all the more than 70,000 papers that had used DNA-based methodologies to assess species or populations. We pulled that. We went through a lot of work in kind of narrowing that down and pulling the information that was actually valuable for our purposes. And we boiled it down to a little more than 600 species, where that had assessments over at least 2 time points, so again, temporal. And looking at those studies and the genetic diversity, we do find a pervasive loss across the globe. But it's not all pitch black. There's also hope. So what our study and findings also showed was that there are conservation actions that can actually mitigate the loss of genetic diversity. So with that kind of positive ending, I'll leave the CBD for now and go into the IUCN. So the International Union for Conservation of Nature, many of you, if not all, have seen the logo before. So it's a huge membership union where you have the Secretariat, you have the Director General, you have the President, you have the Council, but then you also have these 2 large pillars, which is the membership pillar and the Commission pillar. So the membership pillar can be, for instance, companies, NGOs, even governments. And then there is the Commission pillar, which has currently 7 commissions, whereof you might know one of them, which is the Species Survival Commission. And that is also where the Conservation Genetics Specialist Group sits. So under the Species Survival Commission, you can have taxonomical specialist groups, like the CAT Specialist Group, or you can have thematic specialist group, like the Conservation Genetics Specialist Group. And the IUCN, and what it does is closely aligned to the Convention of Biological Diversity. Actually, the IUCN was one of the founding parties of the Convention of Biological Diversity. So these 2 are closely linked. So like the IUCN logo, some of you might have seen the red list logo, both the red list of ecosystems, but also the red list of species. So the red list of ecosystem is basically a risk assessment for ecosystem collapse. So looking at the geographical distribution and the degradation. And the red list of species is the extinction risk of species, looking into metrics such as population sizes over time, historical and current, the census size, the trade, the habitat, etc. But none of these do formally incorporate genetic diversity in their assessments. An example? Okay, an example of that is a recent study by Alicia Mastretta-Yates and colleagues looking at the genetic indicators and red list categories. And there's not a one-to-one correlation. On the contrary, we find that many species that listed as least concern or near threatened have a low headline indicator, so the effective population size 500 indicator. So the least concern on near threat species can experience genetic losses because their populations are simply too small. So none of these have genetic diversity included in their assessments. That's a huge gap. So the Conservation Genetics Specialist Group and a few others put forward a motion to the IUCN pledging that a task force on developing a global standard for genetic diversity assessments was developed. The recently held World Conservation Congress of IUCN is where you do the voting on the motions, and those that pass become resolutions. This motion passed, so it is meaning that it's now a resolution that the IUCN has to work towards. So this is will be a major chunk of work that the IUCN Conservation Genetics Specialist Group has to do over the next quadrennium. So this will be interesting to see where this lands. And it is very timely, not just because we see a genetic diversity loss, but also a recent study from this year by Ivan Pasvinas and colleagues actually showed that around 1.5% of all species assessed under the red list have genetic data. So there's a huge need for generating more genetic data to actually make this feasible. A part of the red list, recent development in 2021 was the development of the green status of species. So recognizing or trying to tell the full conservation story about the species and that are assessed. So it's basically acknowledging the past conservation efforts, but also highlighting the importance of ongoing efforts on conservation for the species, and also set ambitious goals and targets. The pre-impact census size is an assessment that is being done as part of the green status of species. And genetic diversity is often discussed during those assessments. The Conservation Genetics Specialist Group is working together with the team on green status of species to formally include genetic diversity into the green status of species. And currently they're testing that on different species. Another thing that is lacking is the focus on assessments below the species level of IUCN. So only around 1% of all species that are evaluated by the red list criteria are below the species level. So populations, subpopulations are not really assessed. We know that evolutionary processes are local and rely on kind of within species networks on the population level. But people also struggle in terms of how to define populations and the risk of over and under splitting. So the Conservation Genetics Specialist Group has developed a practical framework, but also focusing on the evolutionary significant units. So those that have important genetics in terms of adaptive material. And there are QR codes to the preprints. We are in the review phase, so hopefully it will come out soon, but there's still work to be done in terms of making this a practical tool for people. Practically, can I use 2 minutes more? I'll try to speed up. one thing that we know is the managers struggle with hybrids. So what to do about hybrids? So the Conservation Genetics Specialist Group also had a focus on looking at the value of hybrids, but also in cases where it has detrimental effects. So this is a figure from a recent study that shows the kind of the value of hybrids, but also the opposite end of the scale. And it is a paper that will come out in Conservation Letters very soon. And it is a means to kind of help practitioners in kind of deciding and dealing with hybridization questions. Likewise, we're working together with the Translocation Specialist Group. So they have wonderful guidelines on how to do translocations. Genetic rescue is becoming more and more pressing as a tool. And for that, we need genomic analysis. So we're working on a kind of supplementary to those guidelines to guide people that actually thinks about doing translocations and genetic rescue to incorporate genomic analysis. There is a lot of guidelines being produced and resources material that can help practitioners and others. These are just two of them. So which species to select for genetic monitoring. Wonderful case studies of using genomics, both in Switzerland, but also in California and other places in those guidelines, but also guidelines on biobanking for conservation, outlining standardized approaches to collecting samples, collecting data, storing that, sharing that. There is a gap analysis in terms of what samples are already out there, and outlining a roadmap for a global network of biobanks. So the key messages for today is that genetic diversity is now in kind of the top level. It is in the global biodiversity framework of the CPD and in other kind of higher level commitments, meaning that countries need to report on genetic diversity and use genetic diversity actively in their biodiversity conservation. But also that genetic diversity can be included in strategies and action plans, not just on a country level. It can be done on every level. And there are good examples and suggestions for how to do that. Also, that genetic diversity can be reported on using the genetic indicators, but also that genomic data is key for informing conservation management. The genetic task force of the IUCN is something that will involve a lot of people and expertise in helping to actually frame that. And as we venture into the new coordinating for IUCN, it also means that there will be a revision in terms of membership of the IUCN Conservation Genetics Specialist Group. So if you are thinking of actively contributing to that, have your eyes and ears open, because there will be a call for new members. So first and foremost, thank you for having me on behalf of both Gunnar and I. We're very grateful for having the chance to talk about a little bit about what we do. And a special thank you to all the collaborators and also the ones not on the slide. And for you for listening. Thank you.
Thank you so much, Christina. We only have 5 minutes, but we would like to see some questions.
But there's a coffee break afterwards.
No.
That doesn't work for those online, I know.
There is a coffee break.
You.
Later on, yes,
later
of course.
Okay.
But not right after
Not
that.
right
Yeah.
Thanks, wonderful. Jaakko Pohjoismäki, University of Eastern. I've been involved in some of these red list assessments actually, and was wondering what will be the kind of the practical guidelines in including genetic diversity into those kind of current criteria that are there.
So I know that there's been a lot of work and also a lot of communication with the people, the team involved with the red lists. Currently, they're not that open to actually change that much.
That's my impression, yeah.
Yeah, exactly, unfortunately. And this is also why we decided to go for a task force to explore genetic diversity assessment as a separate, but of course they need to be correlated. And I also heard, I was not at the IUCN World Conservation Congress, but I heard that there were talks about kind of merging things a bit more, at least the species and the ecosystem assessments. So I think it will be very interesting to see where that will go. But currently, genetic diversity in the current red list of ecosystems or species is not really on the horizon.
I see. Thanks.
We have a question online. So Hans Svardal is asking, why should absolute neutral diversity, like a long-term INI, be very informative, useful measure for conservation?
And I know it's imperfect. Let's just say it like that. We would have loved to have many more metrics to actually assess genetic diversity. But the CPD is a multi-treaty convention, meaning that 196 parties, it's a plethora of countries on different levels in terms of resources and investments. So it needs to be something that is a pretty good ballpark and that can work on both non-DNA-based and DNA-based methodologies. But I would love to see many more metrics in there. I would. It's not perfect, and we don't claim that. It isn't perfect. We know that.
Another question here? I have one.
Yeah.
I would like to know, what do you think the ERGA community could do to support not only that genetic diversity is accepted in the different frameworks, but also that genomic data is more rapidly incorporated as one of the core or important technologies that are used for that?
Whew, how long do
we have 1 minute?
So one of the things,
whatever comes from your mind.
And the reason why I showed you the indicator slide is I do strongly believe that we need an indicator using DNA-based methodologies in the global biodiversity framework. It needs to be in there. The team behind all of this have done a lot of work on, interacting with the relevant stakeholders. It's been tiresome, and it's been a huge effort to do that. So it really needs to be direct engagement, and then pledging for something like indicators using genomics to actually assess and raising awareness on the fact that what value genomics actually hold, but also as you know yourself, that there are different resources in different countries. So it's very much also about capacity. So I think a lot of what the BGE does is wonderful, and ERGA as well is wonderful because you already know kind of what are the limitations here, where do we need to put in our efforts. So I think you're doing a lot of wonderful work, and now you need to really push the policy level.
Thank you, and the same for you guys. You're doing wonderful work. Thank you so much, Christina.
Okay, so thank you very much, Christina, for that inspirational introduction to all of the high-level policy work, and maybe how we can begin to feed into it more and more from the ERGA and the BG perspective. And so for our second keynote to kick us off for the conference, we have Alessio from the University of Florence, who's going to tell us a more specific example.
Yeah.
So, floor is yours. You have a, yes, I do. And the pointer is there. There you go.
Is there a pointer? Okay, more or less. Okay, good afternoon, everybody. So today I'm going to talk to you about the integration of reference genome and cytogenomic for biodiversity studies. So in the genomic era, we are all used to think about chromosomes as a series of nucleotides that we usually see in our nice FASTA files, right? But we all know that chromosomes are not just text files that we work with, but they are a specific structure that we have in our cells. They are made up of DNA and protein together, and they form bodies that they have their own specific morphologies, and they are present in different number in different species. And for more than 5 decades, actually, genome evolution was investigated through cytogenetics by looking at these morphologies in different species, as it was very well described in this review from Ferguson Smith, History and Evolution of Cytogenetics. And today also cytogenetics offers scientists a valuable set of tools to investigate the evolution of chromosomes. For instance, you have this nice review in chromosome evolution in marsupials that you see here that was recently published, and then you have the other example on the right about the study of sex evolution and sex chromosomal rearrangement in Sauropsida and mammals that was a pillar for the understanding of evolution of this set of chromosomes in this group of vertebrates. So these 5 decades of data actually has something that we should not toss away, that we should not forget about, because they're offering us a valuable set of information to connect to our genomic data nowadays, okay? But there is this small problem of miscommunication that we have today about the connection between genomic data that you have on the left and the cytogenetic data that you have on the right. I mean, I will explain this with an example. This is Emys orbicularis. This is a European pond turtle species. We actually assemble the genome of this species and we produce the karyotype of this species, which is that one that you see there. As you can see, the karyotype of many turtles, and for sure of all emidids, in those karyotype chromosomes are not listed by size, are not ordered by size. They are ordered by morphology. You first have the metacentric chromosome, then you have the acrocentric chromosome, and then you have the microchromosomes, okay? This is conventional for turtles, and all the cytogenetic data you have on turtles are based on this, okay? And this is what you see on the genome assembly. As you can see, this is an assembly that we produce chromosome level. It perfectly matches the 25 pairs of chromosomes of the species, but you have this small problem that chromosomes here are listed by size. So what is chromosome 7 in our karyotype does not necessarily, I mean, it does not match the chromosome 7 of the genome assembly, okay? So basically this gap make you miss all the data you have gained and until 2025, which is the year we published the genome, based on molecular cytogenetic data, okay? So actually, how can we link these two bunch of data? How can we link cytogenetic and genome? How can we get to the cytogenomic level? Well, actually, you know, we have, sorry, I forgot to mention this review where Lewin, Graves, Steve O'Brien, and all other big names on cytogenomics actually explain very well this problem. So, as I was said, we have some technologies that link the chromosome structure to the genome. We have IC technologies that you know of course, but this doesn't really create a link between the genome and the karyotype. This is just something that tells us how nicely a bunch of assembled genomes interact with each other. So this is something that tells us how to better scaffold our genome, but does not create any link. Well, actually, there is a technique which is single chromosome sequencing that allow us to connect a link between the karyotype and the genome. So single chromosome sequencing is very simple to understand. You basically physically isolate your chromosome, and then you sequence the genetic material of your chromosome, okay? So the sequencing is easy. It's just a normal sequencing in short reads usually. The difficult part is the physical isolation of the chromosome. You can do this either via cytofluorimetry with a flow sorting or with a microdissection if you have enough patients to do that, okay? I will spend just 5 minutes on the description of these two techniques because I think they're not very common and because I think they're cool. So I will spend a couple of words. This is a microdissection apparatus. This is an inverted microscope. So you have objective down there, and then you have a micromanipulator installed on the microscope with two arms. You have a needles, which is used to scratch your chromosome, and you have a pipette in which you collect your chromosome, just like this. So you have your metaphase there, and you have your microneedle. You scratch the chromosome, and then you put the chromosome inside the pipette. And once the chromosome is inside the pipette, you can sequence everything, and then you have your reads of your chromosome. Okay, so this is should be a video that show you how it happens. So you literally scratch the chromosome out of your metaphase. This is a metaphase of a frog. Then you pick your chromosome. They are quite sticky, and then you insert the needle in your pipette, and then in that you broke the pipette into the tube, and then you sequence the material. Okay. You have another method, physical method to isolate chromosome, which is laser microdissection, which is basically works the same with a laser. You just cut the microchromes out of your metaphase, and then you put it into a small tube. As you can see in this other video, you can physically isolate the chromosome with a software. Then the laser cut the chromosome, and it shut the chromosome inside the tube. And in this case, you can barely see the difference, but okay. The other technique for chromosome isolation is the chromosome sorting. This is quite different stuff because it requires a huge amount of cell cultures. So you have to start from a proper amount of tissue source. You don't work with slides. In this case, you work with chromosome suspension. Okay, the chromosome suspension is then run into a flow cytometer. The flow cytometer can isolate the chromosomes based on their charge, which is basically based on their size. Okay. And then the isolated chromosomes are then used to perform a fluorescence in situ hybridization on a metaphase. In this case, the chromosome can be recognized as the chromosome of your karyotype, and then in the end, you have your isolated chromosomes with their names. Okay, this is more laborious than the microdissection, but more effective, as you can understand from this picture. You can gain more copies of the same chromosome and then more material to sequence. Okay. So apart from all of this stuff, the idea is that when you have your isolated chromosome, you perform a whole genome amplification to amplify your genetic material. Then you sequence everything, and then you map everything onto your genome. We usually do this with the DOPSEC pipelines, which are some pipelines developed by Vladimir and Alex at the Russian Academy of Science in Novosibirsk. Now, nor Vladimir and Alex, they're not there anymore. But anyway, the pipeline was developed by them. And so the idea is basically this one. You have your reads, usually short reads of your chromosomes, of your real chromosomes, because you isolated them, and then you have the reads of your chromosomes. Then you have the scaffold of your reference genome, or whatever, how you want to call them. Then you map the chromosome reads onto the scaffold of your reference genome, and then you fish out the information of the reference genome. So by a simple mapping with some refinement, you can find the scaffolds that physically correspond to the chromosome of your karyotype. Okay, so you can find this link, this connection. This is all very beautiful, but how can we use ChromSec data for the study of biodiversity conservation? So ChromSec was widely used to assist genome assembly, and I will show you some example in a minute about this. chromosome sequencing and cytogenomic in general was used to assist genome evolution studies. We have a lot of examples in which cytogenomics was particularly useful for some cases about the study of genome evolution in many vertebrate species. And cytogenomics can actually be used also for biodiversity conservation, and I will show you some examples of this as well. So starting from the first one, for those of you who are working with reference genome, vertebrate reference genome from the very beginning, you may recall this paper. This was one of the first reference genome that came out in 2012. For sure, was one of the first of marsupial reference genomes. And in this case, chromosome sequencing was used to assign 99% of the genome to the karyotype of the species. And as I will show you in a couple of slides, this was particularly useful to identify the genomic region connected with the facial tumor disease of the species. But also, from an evolutionary point of view, the important thing here was that by means of this technique, they managed to produce the first comparative map between the devil genome and the opossum karyotype and the opossum genome, which was the only other marsupial for which genomic data were available. Again, different group of vertebrates, reptile, the green anole lizard was the first genome of reptiles that came out. And also in this case, in 2016, with a different paper, some important refinements were made on the genome to assign the genome to the karyotype. And in this case, these lead authors to understanding some very important, to do some important consideration on the evolution of sex chromosome in anole lizard. And they identified some neo X and Y chromosomes, which could not be identified only on a genomic basis. Now, as you can see, these two papers that I mentioned, the green anole and the Tasmanian devil, were quite old. Nowadays, today, chromosome sequencing is still used to assist genome assemblies of those complicated species. For instance, Archipenseridae, sturgeons are particularly complicated from a ploidy point of view. That karyotype there is the karyotype of Siberian sturgeon, and as you can see, it is octaploid. So as you can see, the metaphase there, it's quite difficult to assemble the genome of such species. And I show you this example here of the sterlet sturgeon genome, which was published in 2020 on Nature Ecology and Evolution. And here a microdissection approach was used to identify homology within the polyploid genome of this species. Somebody with a lot of patience microdissected all the chromosome of the metaphase. Other difficult species are all those species that has a large genome. For instance, the Ambystoma mexicanum has a genome of 32 gigabases. And in this case, the initial characterization of the genome was actually done using a microdissection approach. In this case, was laser microdissection. And authors managed to do some partition of the genome and assemble the genome and try to produce a draft based on data they gained with this technique. Another nice example is this one. This is a paper we contributed to, the pangenome of the barn swallow. In this case, the cytogenetic approach was used to guide IC data. So the IC data is quite difficult to interpret when you have microchromosomes, so in birds, of course, and some reptiles also. So in this case, in the pangenome, we missed a couple of chromosomes that actually were present in the karyotype. So based on the karyotype data, we managed to rearrange, to better curate to do a better manual curation of the pangenome. Last example I'm showing you about the ChromSec to assist the genome assembly. This is the example I showed you before in the beginning. So we perform some preliminary chromosome sequencing analysis of the species that they are already published in that paper that came out a couple of months ago. And as you can see, we managed to microdissect 6 of the macrochromosomes of the species. And as you can see here, as I told you before, chromosome 7 do not correspond at all to chromosome 7 of our genome assembly, but actually is chromosome 12 of our assembly. And this made us do some consideration about the evolution of the acrocentric and metacentric chromosome in emydid turtles based on previous cytogenetic data of the group. As I said, a cytogenomic approach can be used to assist genome evolution studies. I don't have the time now to show you example about the evolutionary studies in which cytogenomic gave a big contribution. I just want to mention these nice reviews here, in which you can see cytogenomic helped the study of genome evolution from fish to reptiles, marsupials, human. Chromosomics is just another term to define cytogenomics. So this science that tried to create the link between the genomic data and the cytogenetic data. But is all this useful also for biodiversity conservation? Well, actually, there are some authors like Potter and Deakin that stated in this paper that cytogenetics should be included in the conservation genetics toolbox. And I think it's something very nice to mention about this technique, is that it can improve species identification and taxonomy. It can help the detection of chromosomal abnormalities associated with inbreeding and disease. And I will show you, and here we will go back to the Tasmanian devil. It can help the study of sex chromosome and sex determination system. And we will see that this is extremely important for species that relies on ex situ conservation program. And of course, cytogenetic and cytogenomic can help also the definition of management units in the so-called population cytogenetics. Starting from this point here, we have this nice example, authors cite this nice example in their review about the wallabies. This species is distributed on the east coast of Australia, and no genomic data were available. I mean, only a little about some markers. And with an integration of cytogenetic data and those little genetic information, they actually managed to define a gradient of, well, in some case species, in some other case subspecies, and in some other case management units along the coast. So in this case, cytogenetic was particularly useful for the determination of management units in the species. Same thing was for these fish in 2 islands in front of Brazil. In this case, no genomic information were available, but authors managed to integrate cytogenetic data, and they actually found two populations of the same species with huge chromosomal rearrangement that made them define 2 different management units. Again, this is the infamous tumor fascial disease in the Tasmanian devil, and by the use of a cytogenomic approach, authors here managed to identify the genomic region involved in this disease, and also that they were linked to sex chromosomes. So again, cytogenomic here was pretty useful for the conservation of the species in this sense. As I said, another important application of cytogenetic and cytogenomics for conservation program are about the understanding of sex determination system. Again, we are with sturgeons again, as 80% of sturgeon conservation program is made ex situ, okay? So it's not made in situ. So they rely, for instance, our Adriatic sturgeon, our Italian sturgeon is now living basically only in captivity, and they rely on the understanding of the sex determination and sex chromosome system to produce a more, let's say, to aid the reproduction of the species. And sturgeon is just an example, but in fish and in polyploid species in general, this is of extreme importance. And not only in fish, in 2022, the microdissection of the Y chromosome of the panda, which is another species that relies a lot on ex situ conservation program, allowed authors to identify some fertility-related genes that were of extreme importance for the design of conservation program for the species. Last but not least, our Komodo dragon, the species that Claudio is studying for more than 20 years, and I'm taking on with the genomic part. The Komodo dragon, I will just spend last 5 minutes, 2 minutes on this point here. So Komodo dragon lives only in Indonesia, in the Lesser Sunda Island. In 2019, we isolated the chromosome of the Komodo dragon using flow cytometry, and we produced the first reference genome for the species using a mix of NGS technologies. We produced a nice comparative map of the Varanus comodoensis with the chicken and the Anolis carolinensis, and we made a lot of evolutionary evolutionary discovery about the sex chromosome system of the Komodo dragon. Especially, we managed to finally identify the region of the genome which is linked to the Z chromosome. And this was quite useful in our preliminary genomic survey of the species in its biogeographical distribution, because we sequenced 24 individuals with a whole genome sequencing approach. And as you know, many reptiles, and Komodo dragon has a ZW chromosome system. And when we came to calculate the coverage for the Z chromosome, of course, we identify half coverage for the females and double the coverage for the males. And this is very important for the species, as the sex marker that were present in the literature were basically non-working or working only with certain kind of samples. But in this case, we managed to identify a whole region that can be used to identify the sex of the species. Last 2 slides. We are now working on a SNP panel that consider only autosomal chromosomes. So we managed to exclude the sex chromosome, and based on this SNP panel, which are 96 SNPs in total, we managed to identify we managed to distinguish all the 6 population of the Komodo dragon very well, and this will be extremely useful, and it's already now to assign the population of confiscated individuals that are present, unfortunately, around. So with this, but this is another story actually. So I conclude by thanking you all for your, with the take-home message. So, which is very simple, that in the genomic era, chromosomes still exist, and sometimes it's worth to have a look at them. And I thank you all for your attention, and the organizer for having me here.
Thank you very much, Alessio, for that reminder, as we are in the genomics era, that sometimes we have to rely on some of our former technologies to see the unseen. Questions for Alessio? You and then you. Yeah. Go ahead.
Can I go first?
Yeah, go first.
Thanks for the, can you hear me? Yeah, thanks for the really interesting talk. As Rob mentioned, I think sometimes cytogenetics is a bit old school, but you showed really nicely how interesting it is in combination with genomics. How big is the cytogenetics community? Because I feel like it's actually techniques you need to, know how to do this in the lab. And I feel like this is a bit like taxonomy sometimes, something that is not so exciting for students. I mean, yeah, can you expand a bit on that?
Yeah, sure. Yeah, well, actually, it's a club. It's a cytogenetic club. You have the cytogenetic club and the more open cytogenomic club, let's say. No, I'm kidding. But it's something that requires a lot of, let's say, skills that are not always written in books, but they are made of protocols that you find from very old professor, then they give you the protocol. And this, but this is true because those protocols react completely different from species to species. So you have to have your own expertise. So, but actually, there is a very active community of cytogeneticists in Europe, in East Europe. They do a lot of this work. Where they actually put a lot of effort also in training students. But well, when I was a PhD student, I did a lot of this in the lab, and I found it quite exciting because you can see a lot of things going on. You can see what you're really working with, and I find it even more exciting when I managed to connect them with the genomic data. So you can see something on the microscope, and then you can translate them into your assembly. And this is, apart from being exciting, I think it's very useful because I found, for example, I mean, I work a lot with reptiles, and for reptiles there is a very old school of cytogenetic, also in Italy actually, and in Prague, in Czech Republic. And I found very useful some papers from the 90s that were just forgot in the literature, and I found it useful to understand some, let's say, connection that I found between birds and reptile genomes that we couldn't explain just looking at sequences. So I think it has room for development.
Thanks. Another question there?
Any information about the application of this approach to plants?
Yes. Okay. No. Plants is a completely different story. I actually had the opportunity to visit some very nice labs which were completely devoted to plants, and they didn't know anything about animals as protocols. And there is this big gap between cytogenetic, animal cytogenetic and plant cytogenetic, because protocols are completely different. With animals, you work with cells, you work with tissues. With plants, you work with I forgot the word in English, with the roots, with the top of the roots. So it's completely different protocol. But in the end, actually, plant cytogenomic was the first to came out instead of the animal cytogenomic. So there was a pioneer group also in Czech Republic where they did a lot of combination also with bionanotechnology. They first combined the cytogenomic and bionanotechnology to scaffold the genome. So actually, the cytogenomic, it's a more common point between the two kind of word, animal and plant.
Okay, I think to stick on time, we should move on. So thank you very much, Alessio.
Oh yeah, why?
Okay, test. And you can sit wherever you want. Okay, wait a second. I need to introduce you first.
Yes, yes.
Sorry. Okay, so we are starting the first case study session. So we will have two presentations, both involving marine species that we actually eat. So this will be more conservation and bioeconomy session. So the first one is Carl Johan.
Yes, perfect.
Perfect. Ruben from Sweden. So he will talk about the mackerel, and we will have after another presentation about clumps. Floor is yours.
Thank you for that introduction. I'm happy to be here to tell you today about how we've been using genomics to inform ourselves about the population structure of two fish species. And I'm here in place of Leif Andersson, but I'm the one who did most of the bioinformatics of the data. So I came here to tell you about it. The two species we've been working with is the Atlantic mackerel and the blue whiting. And the Atlantic mackerel is harvested by fisheries to a volume of 800,000 tons annually. And the blue whiting, which although less known, is harvested at about twice that amount. So as you can tell, they are economically important species. And our key questions and goals with this project is to use whole genome resequencing using Illumina to enable the exploration of population structure and the identification of loci that are subjected to genetic differentiation between subpopulations or stocks of these species. And the first question we ask ourselves is how divergent are different populations of these two species? Second question, can we use these models to gain insights regarding the genetics of ecological adaptation? And one of our hypotheses is that some divergent loci represent loci that have responded to the ecological adaptation. So the way we tackle this is to sample about 30 individuals. I'm starting with mackerel. So whenever you see this striped fish, I'm talking about mackerel. And whenever you see the blue fish, I'm talking about blue whiting. And you see the dots on the map. That's where we sampled. And as you can see, one of the populations we sampled was from the North America, around Canada. And that's called the Gulf of St. Lawrence. And it's the last population in the table over there. The other populations are scattered across Europe, and one of them in the Mediterranean Sea, some quite far north in Europe. And the first, and to our surprise, what we saw when I started analyzing this is that the Algarve population clustered very peculiarly in the PCA, and it was because it wasn't Atlantic mackerel, it was chub mackerel. So we threw it out. I will tell you how we analyze this in the coming slides, but first I will tell you about blue whiting. And as you can see here, we had a very similar strategy. We sampled individuals across the distribution range, here mostly around Europe. We also created Illumina libraries here for about 240 individuals, with 20 to 30 individuals per sampling location, aiming for a WGS sequencing depth around 2 to 7x. We're actually aiming for 5, but this is the way it ended up being. This is our analysis pipeline. We isolated blood using QIAGEN blood and tissue kits. We made custom Illumina libraries using in-house custom TM5 protocols, or a quite new method developed at the National Genomics Infrastructure in Sweden that they call Upsplat. Giving quite cheap Illumina libraries because we sequenced about 500 individuals. We wanted it to be as cheap as possible. We sequenced on Illumina machines at NGI Uppsala. We analyzed the data in one of the Swedish supercomputing clusters. We aligned using BWMM SAM tools, did the duplicate removal, did 2 types of SNP calling, freebase, and also direct SNP calling in angst. And to infer population structure and FST, we used angst and PC angst. And we ended up getting this type of information for the mackerel. In the colorful table to the right, you can see the pairwise FSTs, with the highest pairwise FSTs depicted in red. And that's St. Lawrence and Catalonia. So the Mediterranean population and the North American population seem to be most divergent. And you will also see this when you look at the PCA, where you see that the red dots, which is the Gulf of St. Lawrence, they differ quite a lot from the rest of the populations along PC2 axis. And so does the Catalonia population, the Mediterranean population in blue. And when we show the FST in the Manhattan form in the lower right, you can see that this differentiation between these two most divergent populations isn't just in a few loci, it's all over the genome. So we have a lot of differentiated loci. And now we switch to blue whiting, and here we got a completely different signature. So we first we go to the lower right PCA, which is the PCA of about half the chromosomes look like this when you look at them in PCA. So you have a clear outlier population, which here is the Mediterranean population in light blue, clearly different from everyone else. The two Icelandic populations, which are light pink and red, they're also slightly differentiated from the rest of the main clusters. But if we go to the second PCA, about half the chromosomes look like this. They position themselves in 3 different clusters around PC1. And we believe this is due to massive inversions on many of the chromosomes. And what's also commonly seen for these putatively inverted regions is that the Mediterranean population sticks out like a sore thumb. And when we compare the Mediterranean population in Iceland in the Manhattan form on the top, we can see that the divergence actually exists in quite huge clusters. For some chromosomes, like this 44 chromosome, covering more than half the chromosome. Just going a bit further on this topic, here we show, I show on the mid-lower FST plot, we see that more than half the chromosome that ends with 37 is highly differentiated between the Icelandic population and the Mediterranean population. And this means that we have a quite huge amount of highly differentiated SNPs in this species, some which seem to lie or reside in massive inversions, and some that don't. And altogether, we have identified this type of regions, and these can be used for various purposes. One of these is to gain insight into the genetic mechanisms behind ecological adaptations, but obviously also to make informed decisions in fisheries. So we identified the most relevant stakeholders to be the European Association of Fish Producers organizations. And we know also that European Union advisory councils are involved in decision-making. And so is the ICES, the International Council for the Exploration of the Sea. And out of these, it seems like the ICES and the European Commission are the organizations that will consume the evidence we produce to possibly alter policies for fishing. We will deposit these datasets in open repositories following a bit more data exploration because we're not completely done yet. And then we plan 2 publications at least. First, an Atlantic mackerel paper, and next, a blue whiting paper. And the reason we want to go with the blue whiting paper later on is that we find these putative inversions very interesting, and we want to do assemblies, including high-C and PacBio assemblies of Mediterranean populations, to be able to actually observe them in data and not just through genetics methods. And the next steps, which we already embarked upon actually, is to put the divergent markers and neutral markers on SNP chips. And we already have used SNP chips like this. And to the right, I link to a paper by Leif, showing how he and his team has used multi-species SNP chips of about 2,000–3,000 SNPs per species to enable rapid and cheap genotyping of populations from fishing vessels. And I ran a script that the guy in the lab, Mats Pettersson, has developed to identify the most divergent SNPs exhaustively. So you can basically trust this script to basically identify the most informative SNPs in the data. So once it's, for example, exhausted the information from the Mediterranean populations, it switches to the next. So now we have about 3,000 SNPs that we will be available for those who want to do further research, which we also plan to do, but also that can be used for fisheries management purposes. And that's it for me, and I would be happy to answer questions.
So we have some time for questions from the audience. Camilla?
Thanks a lot. I really like this work. I think it's a perfect example for us to showcase. I was wondering, when you showed the chromosomes patterns with possible inversions and so on, am I right to say that these were high numbered chromosomes? Are they the microchromosomes that you're showing, or this happened across
happened across size ranges? Okay. So this particular one is the second largest chromosome.
Ah, okay. But did you see a pattern between macro, micro?
Not really.
Size related? No.
So some of the quite small ones had this pattern, and some of the really large ones. So I think like the second largest, the fifth largest, the 10th largest, it was scattered all over. Okay. I didn't see any such pattern.
Yeah. Okay, good. Thank you.
And we have a question from online. How expensive is it to develop a SNP chip, and is this done in-house or by commercial partners?
Yeah, I wish I actually had the answer to this particular question, but so this is not in-house. So it's a company. And the reason we do this as a multi-species chip is to keep costs down, because then you can order larger quantities and the design is already done. Yeah. But assuming you only need a couple of thousand markers, obviously the resolution is worse. Yeah. But I suggest you email Leif Andersson, because he will be able to tell you.
Yeah. Yeah. And I mean, the good thing about this multi-species SNP chip is that they don't necessarily have to be all fishes, but you can have like fish, birds, and stuff like that. So
yes,
it's relatively cheap. Okay, yeah, one last question from the audience.
I have a loud voice, so maybe you can hear me. Okay, sorry. So thank you for this presentation. It's really interesting for the one working with commercial fish, so which is tightly linked to management. And do you know if those inversions, so the group characterized by this putative rearrangement, do they have also difference in life history traits?
It's a very good question, and we just don't know yet.
Okay.
I just showed you one example here, and as you can see, all the Mediterranean populations are out here. So the Mediterranean is the most extreme. They only have one version. But if you go to the other ones, it doesn't really always look like this. although the Mediterranean one is the most divergent. So we believe that we actually need to sample more populations in order to get a better grip of why this is. But for those who are into inversions and fish, for example, the cod.
Yeah, that's what I was
which is also
thinking.
relative to the blue whiting because it's a galliform. The scray cod, which is a very prized cod for food, differs from other cod populations for a large inversion. And here we have the same pattern, but for many different chromosomes, and it's super interesting for further exploration.
Okay, perfect. Thanks a lot, Carl, for the presentation. And we move on to the last one of the first case study, which is by Enrico. The microphone?
Yeah.
He went away with the microphone?
Now it's not.
Otherwise, I can give you this one. Oh, okay.
Everyone does that.
Yeah, I didn't know that you also had a microphone, sorry. Okay, perfect. Yeah. Okay, perfect.
Just this one. Yeah. Okay. Good afternoon, everybody. I'm Bortoletto Enrico from University of Padova, and today I will present our work on developing a chromosome level assembly for Camellia gallina, basically to assess the exposure risk associated with microplastics. The problem, basically the most important species for farming in Italy and I would say Mediterranean Sea was Ruditapes philippinarum. But nowadays, several factors are impairing the production of this clam. First of all, heat waves, especially if you think about lagoon environment, like in the case of Venice. In the lagoon environment during summer, the temperature can reach really, really high level, like 30, 35, even 40 in some days, okay? But the major concern is Calinectes sapidus, so the blue crab. Blue crab is basically eating all Ruditapes philippinarum. That's why Camellia gallina can represent a good alternative to Ruditapes philippinarum, because Camellia gallina live more far from the coast compared to Ruditapes philippinarum and burrow deeper in the sand. Since the blue crab is quite lazy, so they can survive. So we started to study this species, and in our first study, we wanted to investigate the interaction between Camellia gallina and micro and nanoplastics. The questions were, does Camellia gallina accumulate micro nanoplastics? So is it safe to eat Camellia gallina? And secondly, if this micro nanoplastic can alter the clam physiology. For the human exposure part, basically we just sample Camellia gallina around the Mediterranean Sea and quantify with chemical methods the amount of micro nanoplastic inside the clam. But for the physiological responses, we planned to expose Camellia gallina to micro nanoplastics for 30 days and then study possible alteration in transcriptomics and methylation patterns. But no available genomes for Camellia gallina. So the first step was to build a reference genome. And also basically no sequencing data, just 16 RNA-seq dataset and 1 transcriptome. So supported by ERGA, we started to produce the genome, and simply producing IC and PacBio reads, we use the classical pipeline, IFIASM and scaffolding with salsa young closer, and then manual curation. As regards the genome annotation part, first of all, we annotated the repeats in Camellia gallina. Then with Breaker, we obtained the first gene model draft, but also we produced some RNA-seq dataset to support Breaker, but also with Trinity, we assemble a transcriptome and integrate this information in our gene draft with PAS and evidence modeler. For non-coding RNA, we just use Infernal, so simply. Obtaining, in this case, a final annotation, we also try to validate further this final annotation with the nanopore reads of RNA, but we use a mini ion, and basically the coverage was not super good, and we can validate only 10% of the gene with nanoporids. Some statistics, basically the genomes is 1.8 gigabase, and the coverage was 41x with the I5 reads, and 97 million, so quite results. Also, we checked for contamination with the blob toolkit, and we didn't find any major contaminants. Busco values quite good. For example, in metazoan, 95.6%. But also, we tried a new tool. This tool was applied for human pangenome project. Flagger can estimate the correctness of a genome assembly based on the coverage. I don't know how actually it's quite complex, the algorithm. But running Flagger, we obtained a value of 99.75. That is quite good because in human, they obtained 99.2. So,
wow.
Wow. Super lucky, actually. Super lucky. Because then in muscles, different values, actually. But yeah. So we obtained a high quality chromosome level assembly with the 19 chromosome matching this nice karyotype produced by a Spanish group. So everything was okay. But there are some limitations. In bivalves, we have a lot of repeats in the genome. In this case, 54% of the genome is composed of repetitive elements. And I localized the error I made, estimated by Flagger, the erroneous region, and 71% of them are in unclassified repeats. So maybe centromeric or satellite, I don't know. And then the other problem with bivalves is that our group, we realized that, for example, Mytilus gallo-provincialis is characterized by an open pangenome. So just one individual is not enough to capture all the variation. In this chart, you can see basically 16 different Mytilus gallo-provincialis. So each circles represent a different individual, and also genes that are missing in that specific individual. So basically 30% of genes are in pangenome in Mytilus, a quite extreme case. Okay, moving to the stakeholder engagement part, we collaborate a lot with the IZS Teramo, basically Italian National Health Service. Then I participated in a project in which we have to prepare some university-style lessons for high school students. And during these lessons, I basically showed the ERGA project and also explaining why it's so important to extend the repertoire of reference genomes. We worked with the mollusk farmer consortia because they are quite interested in our results. Also, we deposited some samples in a museum for biobanking, but also some shells because this is a museum full of shells of Bibles. And also, I did a poster presentation at the Mollusk Farmer annual meeting. For policy, I would say quite early for that, but in the future, EFSA could use this result to support, basically to say, yes, it is safe to eat the Camellia gallina or not. We already presented this case study at the Micro 2024. IZS Italy, so the National Health Service of Italy, because they can use this as a tool for seafood traceability mainly. Fisheries, also this is quite futuristic because for the moment, no genetic selection are made for Camellia gallina, basically not for bivalves, with the exception of Crassostra gigas, the Pacific oyster that they started to. And of course, this genome can be a good resource together with all the genome produced in the ERGA BG project, showing that assembling genome is not just fun, because it's fun for me, but can also be super useful for the scientific community. As regards the impact, actually unexpected results, because in LinkedIn I filtered, thanks to the dashboard, the ERGA post, and I obtained in 1 year basically more than 7,000 impressions. And one of the most famous posts was the one about ERGA blog post, showing that that was a really nice initiative. all the data are now included in a paper that has been accepted by scientific data. And the next steps, of course, publication, because we are waiting for the proofreading, and dissemination of the paper through the established communication channels. They asked for the Genoma group, asked for our genome, because they are doing a population genomic study on Camellia gallina and Camellia striatula, so another bivalve. And finally, we plan to strengthen the stakeholder engagement and to try to generate additional DNA-seq dataset to develop a pan-genome framework also for Camellia gallina. To conclude, I want to thank you, ERGA, BGE, University of Padova, my institution, Plastic Bomb Project, and colleagues from IZS at Erasmus.
Thanks a lot, Enrico. We still have time for a few questions from the audience, if anyone has one.
Thank you for the talk. I didn't quite get the point you made with the pangenome of Mytilus. Does it mean that you have gene presence absence in different individuals, and how do you detect that? Because you have only one reference,
Yeah,
so should we
actually,
do population genomics of chromosome scale? You know, could you expand a bit on that?
This is not my story, because this story started, I think, 10 years ago by a guy working in Trieste, Marco Gerdol. Okay. And basically, they realized that I don't have the chart, but my colleague Umberto last day will show the chart. In the coverage map, they have a super big peak haploid. So 1N and 2N. So super unexpected, no? You know, the classical genoscope profile, no? Super unexpected profile. So they start to testing different theories. At the end, they developed a bioinformatic pipeline that basically exploiting the difference in coverage. They showed that some regions are characterized by just half of the coverage compared to rest of the genome. So like sexual chromosome, something like that, no? But actually was gene. Then now we are assembling actually 12 different genomes of Mytilus galloprovincialis with so like that, IC, I5, and so high quality chromosome level genome. And we are seeing this type of, also in this data, the presence substance variation, basically. And also following Mendelian law. So you have just, for example, we're planning maybe to do some cytogenetics, because you could see with the right probe a chromosome with one chromatid with the gene and the other one without. So it's like strange, really, really strange adaptation.
Well, for the sake of time, we have to end it here. There are some questions for you online. So if you want, maybe you can go to YouTube and then maybe answer the question. And well, so we close here these first sessions. We're going to have the coffee break, and then we will have the second case study sessions. So thanks a lot for all the presenters. Microphone.
Microphone.
Thank you very much, everyone. We will start the second and last case study session of the day with 4 talks that are here to remind you that there's much more to biodiversity than what's cute to look at or what you can eat. So for the speakers, I'll warn you at 10 minutes, and then again at 12, and at 15, I'll unplug the TV and we'll move on to the next speaker, okay? So first up, Olivier. So there's like algae. Thank you.
Just a second, trying to figure out. No. Okay. Yeah, welcome everyone. Olivier de Klerk from Ghent University. I'm gonna present a case study that is focusing on seaweed. Now I realize seaweed may not be the center of everyone's world, but this one you can eat. And actually, yeah. And actually, we didn't rehearse this, okay? Actually, this one we do eat. Good. So I'm going to talk about Porphyra. And what is super cool about Porphyra, that not actually Porphyra itself, but related genus is cultivated at a very large scale in Asia, and talking China, Japan, Korea. And it's one of the very few seaweeds that has actually made its way into our Western lifestyle, the way we eat. Now it's very trendy to go out and eat sushi or buy it at a supermarket. Now, if you wonder what Porphyra is, so it's that black wrapper that very often surrounds this maki or this sushi. Now, I'm not here to advertise that you have to eat seaweed, obviously, but I would like to stress, though, that the success of Porphyra cultivation in Asia, it's actually the success of controlling the life cycle. And Porphyra has a quite complex life cycle, whereby we have 2 phases. We have a haploid phase and we have a diploid phase. Now the haploid phase, that's the blade, that's the crop. That's what you want to harvest. Now the diploid phase is very different. The diploid phase is a small filament that grows inside, usually inside bivalve shells, in oyster shells. That's where it's commonly found. Now, the transition between these 2 life cycle phases, it is key to its success as a crop in aquaculture. Apart from the sexual cycle that I just kind of explained, there's also an asexual cycle. Now, what the Asians managed to do is actually combine that sexual and that asexual cycle. The sexual cycle obviously generates the genetic diversity, and the asexual cycle, once you have a very good genotype, let's say, for cultivation, you can replicate it very quickly. So it's these 2 aspects that are important. Now, why am I talking about Asian seaweeds? Because of the European Commission and the whole blue economy aspect of it is that our future will incorporate eating more seaweed or using more seaweed for various purposes. And one of these aspects that we could actually hopefully introduce into Europe is actually the cultivation of Porphyra. So there's 4 species in Europe, but there's claims, serious claims, I would think, of cryptic species, of hybridization, and also of ploidy variation. Here you see from a number of papers from, that was not the idea. I screwed up, sorry. Okay, so this is a number of illustrations from Varela. She's a researcher in actually here in Portugal that has been looking at Porphyra diversity quite extensively, where she has Porphyra dioica and linearis, and these are the 2 species that we're focusing on here. But these hybridize, and then within this linearis, you do have probably cryptic species as well.
The middle panel, I can't point, this flow cytometry results that demonstrate that there is diploid, tetraploid lineages in this linearis, and on the right-hand side, it's a structure plot where the ploidy level of some of these goes up to 8x, so lots of ploidy variation in there. Good. So in terms of what we're trying to do here with this case study is, well, we're actually trying to build a genomic resources that we can demonstrate this reticulate evolutionary history of porphyra. That's what we're trying to do. And secondly, if we want to see if asexuality is part of that life cycle, whether that can actually be linked to hybridization and polyploidization. What if these asexual lineages that we also find in Europe, I mean, these might be very weird ploidy levels that just recycle themselves and do not enter the sexual cycle anymore. Good. So what is out there in terms of genomic resources? Barely anything, I would say. So porphyra has got 53 species worldwide. It's got one genome published, which is a Northwest Atlantic strain, so from the US, of Porphyra umbilicalis. That's a picture of it. And it's a small genome, which is fantastic, 87 megabases. And they managed to publish this with 23,884 contigs. So highly fragmented, even though it was supposed to be PacBio. But it's already, let's say, research from about 10 years ago. So what we're trying to do is build chromosome level resources for two of these European species, Porphyra dioica and linearis. Here you see a bit the flow that we're using, which does involve nuclei isolation to get rid of mainly bacterial contamination. Then we did high C on that, we did nanopore reads and hi-fi reads on that. And then for dioica, I think the results are still quite recent. We do arrive at a genome size of about 140 megabases, and let's say between 8 and 10 chromosomes. So that looks quite successful. Do note that in this high C, this juicer map, there is still quite of bacterial contamination that plays there. Linearis tells us a completely different story so far. So where we have a genome that is a lot larger, 350 megabases, possibly diploid, polyploid, things we didn't expect. And there's definitely a need for a lot of post-processing that is going on over there. This juicer plot was not what we were expecting. So there's things to be done there in terms of the bioinformatics, and hopefully we can tease out its haploid genome there. If not, we'll have to redo the sequencing, I think. Good. Challenges. The reason that there was only one of these Porphyra or Pyropia genomes out there, I mean, sequencing seaweeds is not very easy. These things live in the sea, they contain a lot of bacteria, and bacterial contamination is a big issue. I've seen one of these plots before from blob tools, and well, everything is bacterial except this. Okay, that's Porphyra. So I mean, we're very good at sequencing bacteria, and then trying to filter out the reads. Second thing, and that's actually linked to the bacterial contamination, is that especially in ONT, so nanopore sequencing, is that DNA of seaweeds and plants tend to clog these things. It's not fun, because then in the end, you just tend to sequence bacteria. Good. What is the long-term vision? The long-term vision is actually to produce these genomic resources of the European species, and then combined with culture experiments and flow cytometry, then we're going to make a marker set and then design a high-plex PCR pipeline or flow through that we can easily screen this Porphyra diversity at a population level. This one is part of a publication that is just coming out where we do find that this sexuals and asexuals, I'm not sure if it's clear from this map, that there is both a geographic division there, but even on very small scales, that's where these porphyras occur. For example, here on these boulders, we tend to have sexuals and a bit of asexuals at lower parts on the shore, and asexuals in the more stressful environments higher up on the shore. So these are the things that we are looking into there. Stakeholders and engagements. So far, we've been focusing to present the results and the ideas that we have in a project that's called GAME. It's a research project, strategic basic research from the Research Foundation in Flanders, which has a very strong industrial advisory board. So for people who want to use porphyra biomass either for food or for other valorization purposes, such as the chemicals that it contains. Next steps. So, well, this linearis genome still needs a bit of work, or a lot of work. And then we also want to definitely generate a chromosome level genome sequence of Porphyra umbilicalis. So, and then perhaps hopefully try to interact with the people from the Darwin Tree of Life, because they've been sequencing Porphyra purpurea, and then design this marker set of for population level genotyping. Good. Dissemination, publication, hopefully, if that would work, would be very nice to have a genome paper that reports on the genomes of 4 European Porphyra species. And then the other 2 points, the population level sampling and design for crossing designs. This is actually part of the PhD projects of Jordi Morcillo and Silke Boekenhoven in my lab. And these are these receive funding from the Flanders Research Foundation.
You'll need that still. So we have time for one long or a few short questions.
Thank you very much. Yeah, good luck disentangling that. With the ploidy situation and the hybridization situation, are there kind of special factors you have to take into consideration designing the kind of diagnostic marker selection for species population discrimination when you have all of this ploidy going on and all of this hybridization going on? Like, will you be able to tease them apart?
I'll answer in such a way that people have done that on other groups. Yeah, ferns, for example, they have got a very good track record of making that realistic. Where we made an error is in selecting our Porphyra linearis strain. Yeah, so I think what we should have done is done the flow cytometry first, and then go for the lowest genome size and start from that. But as it happened, we picked another strain.
Good luck.
I wondered if I could also ask, does this have a stable karyotype, and particularly with these sexual and asexual versions?
I was very amazed by the talk about the chromosomes. In seaweeds, we know very little about chromosomes. When we check the literature, they range from 2 to 12. So when we arrive at something like 9, but the diploid haploid thing, that's normal. Yeah. That's normal for plants and seaweeds. Okay.
Thank you very much. Okay. You two are going to speak in tandem, parallel. Yeah. Great. Looking forward to it.
We'll make some show.
You'll make show. Perfect. Thank you very much. That's the big one.
The big one.
Yeah, yeah.
Okay. So hello to everybody. I'm going to tell you now, we are going to tell you how integrative taxonomy and involvement of farmers helped us to trace new non-native oyster pests in the Mediterranean Sea. And the stakeholders had really big importance in this research, not only as users of our results of our research, but really as active participants in the research. So that's why we invited here also the representative of stakeholders that will present together with me the second part of research. So aquaculture is one of the main vectors for the introduction of non-native species in the Mediterranean that can be introduced whether as target species or hitchhikers on these species. So here you can see, for example, one Pacific oyster with very rich falling fauna that can be eventually introduced together with the oyster in some new area, because translocation of both juvenile and adult mollusks is very common practice in mollusk farming, contributing to the transport of non-native species. Unfortunately, both national and European laws are not adequate for to manage that problem, because they are speaking only about transfer of non-native species as target species. Nothing regulates transportation of hitchhikers. So that's why we should overcome that problem in other ways, and that's why involvement of different stakeholders like farmers and scientists and also legislative and management bodies is very important. So oyster farming is one of the principal aquaculture industry in the Mediterranean Sea, facing really numerous environmental problems. One of them is also the infestation of oysters by these pests, worm pests, of the genus Polydora that bore into oyster shells. And here you can see that they make kind of mud blisters that are really ugly to see, smelly, also smelling on sulfurs because of the degradation of organic matter inside, and also making oyster shells very soft, which is a big problem for marketability of oysters. And many oyster farms unfortunately failed or have really big economic problems. So knowing which species of this genus we do have in oyster farms, and also where they're distributed, is very important for their management. But many of them are representatives of species complexes. So in order to determine them correctly, we need to have to work both on morphological and molecular methods. So the objective of our study was both to confirm the identity of these worms in oyster farms in the whole Adriatic basin, and to understand their distribution, both based on morphological and molecular analysis, and to try to clarify the provenance and way of the introduction in the basin. For that, we should use reference genome and genomic data that should contribute to answer to these questions. And also, we aim to strengthen connections between different stakeholders for the successful management of these pests. Our principal questions were, are there non-native Polydora species in oyster farms in the Adriatic Sea? Are different species maybe distributed along two Adriatic coasts? Are there farming practices that farmers do that might contribute to their transport? And how did these species spread in the basin? So to answer all of these questions, we really need interdisciplinary and transborder research, because we wanted to speak on the basin level. And that's why in this research are involved both University of Bologna from Italy, University of Dubrovnik from Croatia, and also representative, the Cooperative Mare, cooperative for the research and support to aquaculture industry from Italy. So we sampled oysters in 11 oyster farms along Italian coast of the Adriatic Sea, and 6 oyster farms
along Croatian coast. We sampled 15 oysters per farm, put them in low salinity solution in order to stress polydoras and make them come out from their holes, and then conserve them in 96% ethanol, did morphological analysis that are really long and difficult, but are necessary. Unfortunately, not all of them, we were not able to determine all of them morphologically, so we need to use also molecular identification. We sequenced several markers. Then, of course, we did new sampling, because samples had to be taken in different way and handled different way for genome sequencing. And then we started to work on population genomics. So what did we find there? Our molecular identification indicated that we have two new non-native Polydora pests in the Mediterranean Sea invading oyster farms, Polydora websteri and Polydora sica. We already published that research. Very interesting thing is that we have different composition of Polydora species, different species living along Croatian and Italian coasts. So Polydora sica and Polydora websteri are distributed only along Italian coast, so only in Italian oyster farms. And that's really interesting. And the situation is sometimes really critical, because that's what we can find. Some of the oysters are super invaded and very fragile. So why all this matters? Because we indicated to stakeholders that they actually have new pests, non-native pests in their farms, that monitoring measures should be started. This also implies that we should do probably new pest control strategies, because we do have new species that probably will not respond to usual treatment measures for your oysters as the other species. And the insight that we have different species along two coasts opened some new questions. So why is it like that? Because different species are farmed along Italian coast, where I forgot to tell, it's a Pacific farm, Pacific oyster in Italian farms, and European oyster is Croatian farm. Is that the reason? Or different translocation practices, or different treatment practices? So that's some question for the future. Unfortunately, genome sequencing was not successful by now. It was very difficult to start to work on population genomics. We did hundreds of extractions of DNA to got enough of the DNA to be able to do RAD sequencing. So we started just now to do DDRAD. So results of our research indicated some very important surveillance questions. We should monitor Polydora websteri populations. We should do a survey on the efficiency of usual treatment methods. And the most important is that surveillance of mollusks transfers should be done, which is the biggest problem of all, of course, because there are no legislations, and sometimes farmers do not say that they grow an alien species or that they do transfer. So these are all very big problems for management. And we can overcome these problems by educating farmers, transboundary collaboration between countries, and also, of course, by including different stakeholders to solve this problem. So now Eugenio will tell you a little bit about stakeholder engagement, because he was personally working on that.
So in 2024, we did a survey with farmers along the Italian Adriatic coast, in particular to 24 mussel farmers and 10 oyster farmers. And the goals was to understand the knowledge of the farmers on non-native and pest species, and also to gain some information on the translocation of the mollusk for farming purpose. And also during the interview, we were able to educate a little bit the farmers of this topic, so non-native and pest species on the farming system. And as you can see, we have really interesting results. We created this network analysis map based on the answer of the farmers. And as you can see, there's a lot of movement and translocation of mollusks for farming purpose between Italian farmers and other Italian farmers, between Italian farmers and other countries, Spain, Greece, and France, and also inside the Mediterranean basin, but also outside the Mediterranean basin, the Atlantic coast of Spain and France, for example. So this underline that, of course, this movement can contribute to the transportation of non-native and pest species from different farms. And so we have to do something to manage, to be able to manage this and improve, of course, our knowledge and try to stop this movement and this problem non-indigenous, non-native and pest movements. And in 2024 and 2025, we participated in a conference, the Serum, Italian Societies of Research Applied on Mollusk Farm, where we have workshop with all the stakeholders. As you can see, that a lot of stakeholders from different levels, so from farmers to ministry, Italian ministry, veterinarian offices, Italian Ministry of Health, Regional Environmental Protection Agency. We talk about the BGA project and the case study that we have and all the results and activity that we do during the project. And we also were able to talk about the future step that we have to do for the management of non-native and pest species in farms. We also have an impact dialogue. We have an impact dialogue with the University of Bologna, in 2025, talking about these topics. And okay, all the results of our project will be used to both for European and national strategies aimed for the containment of non-native spread through aquaculture, for designing of surveillance protocol, both on Polydoras, of course, but also for other non-native species that can be transported through oyster aquaculture, to create guidelines for necessary education of activities for farmers. We have to educate the farmer if we want to use the farmers to, if you want to use them for what they know about their farmers and the non-native species that they do see in their farms. And for the future, so we have to organize more meetings with stakeholders. We have submitted a joint project in a regional call, Emilia-Romagna call for project with the stakeholders between the University of Bologna, Mare Cooperative, and 2 oyster farmers about the development of protocols to remove Polydoras from the oysters. And also we are now trying to create guidance leaflet on non-native and pest species for mollusk farmers, so with images and description of the non-native species that they can find. We already published an article about the 2 new non-native species, and one is under the revision and another in progress. And that's all the people that we reach with our case studies. So 100 representatives, oyster farmers in Croatian, Italian, also high schools and other universities. Thank you.
Short question.
I thought that we don't have time, so escape.
Just what sort of language do you have to use to talk about? Do you talk about genomics, or do you just talk about
with the farmers?
Yeah.
No, of course. No, we only talk about the identification of the species in general, so which species they can found in the farms. they show us through videos and images what they found, and what they found like weird. Oh, this is new for us. We never see that before. So there's an exchange of information about new species that they found, and can be impactful for the farmers.
Small correction, we did speak about genomics because we did a survey with stakeholders on both conferences, and then they could hear about genomic research when we presented the presentation on the workshop. So yeah.
A little bit. Yeah.
A bit, yes.
Thank you.
Sorry. So I'm going to use
I'll call it Eleanor at lunch, who asked Murray, what's the point of mosquitoes? And I wondered how many fields you can ask this to someone's face.
But yeah, I gave a very good answer to that. They are also pollinators, for you, those of you who don't know it. So obviously my talk is going to be about mosquitoes, and in this case it's going to be about the bad part of the mosquitoes instead of the pollination. It's going to be how can we use genomics for vector control, and I'm going to focus on Culex mosquitoes. So how did this project started? So basically West Nile virus is one of the most important emerging infectious diseases we have in Europe since 2010. It has spread all across the continent, and we have outbreaks every year in different countries like Italy or Greece. In Spain, until 2020, the cases were very rare, but then in 2020, together with COVID, just to make things a little bit spicier, we had our first outbreak of West Nile virus that has been followed by a second outbreak in 2024. So what happens with West Nile virus? West Nile virus is a vector-borne disease. It's transmitted by mosquitoes. More than 60 different species of mosquitoes can transmit the disease. Most of them belong to the genus Culex. Birds are the reservoirs, and occasionally infected mosquitoes bite humans, and then humans get infected and can get the disease. So what happens in our area, southern Spain, in Andalusia, which is the area where all the outbreaks had happened in Spain up to now? We have 5 different Culex mosquitoes species that can transmit West Nile virus, but 2 of them play the most important role. Culex perexigus, which is the one that mostly amplifies the cycle in the birds and usually inhabits the rice fields, and Culex pipiens, the house mosquito, the common mosquito, that acts as a bridge vector that can transmit the disease to humans. So what we wanted to know is, why are these mosquitoes different? We had no information on Culex, on this species of Culex mosquitoes, no genetic information, and no genomic information. Anopheles has been studied for a very long time from a genomic perspective, but when we started this project, there was only one Culex genome available, Culex fasciatus. So the objective was to use comparative genomics and population data to uncover a little bit the evolutionary mechanisms shaping vector capacity in the Culex mosquito species that we have in our area in southern Spain and are causing the West Nile virus outbreaks. And to do that, we sequenced 4 new genomes of Culex mosquitoes. And we divided the project in 2 main questions. One had like a more evolutionary questions using comparative genomics. We are studying ORAM binding proteins, olfactory receptors, and gustatory receptors to understand how they have evolved in the species, because they have different preferences for their prey. Some of them prefer to feed on mammals, some of them prefer to feed on birds. And then we also wanted to know from a population genomics perspective, both what is the population structure and insecticide-resistant patterns in Culex pipiens and Culex perexigus, these 2 species that, as I told you, are the ones that are driving the the West Nile virus cycles in Spain. So we sequenced 4 genomes thanks to Sanger and the work of people like Tom. We have the reference genomes for Culex laticinthus, Modestus, Perexigus, and Telleri. We assembled 3 pseudomolecules, and the boost completeness was between 90.6 and 95.2%, and the scaffold N50 ranged between 150 and 292. For population genomics, what we did was mosquito captures that we sampled at 25 different sites in Andalusia. We genotyped 217 Culex pipiens and 133 Culex pelexicus. We identified first the visu, each species, and then ran barcoding. Then did whole genome sequencing and SNP calling using VCF tools, Blink, and cleaned all the data to identify both population, to do both population structure analysis and do identification of inversions, because we have in one of the species inversions for them. So I'm going to go into the genome insights. These are preliminary data using the comparative approach. So these new Culex genomes are now, these 4 new Culex genomes are now 50% of all the genomes available for Culex. So right now we have only 8 genomes available, and only 7 of them are annotated. The Culex pipiens molestus is not annotated. This reveals some preliminary patterns in chemosensory gene evolution, and I don't know if you can see the little numbers, but here, these 4 species, which belong all to a complex called the complex Culex pipiens, have more odoran binding proteins, odoran receptors, and gustatory receptors than the species that are not from the complex Culex pipiens. One of the hypotheses that we might start working on is that Culex pipiens and all the complex in those species are very generalist when they are feeding. They prefer birds, but if there are mammals like humans, they'll feed on mammals. However, the other species have are more specific. Culex teyeri only feeds on mammals, and the other mostly feed on birds. Unless there is no prey available and they are going to die, then they will feed on what is available. But Culex teyeri, for example, even won't take a bird bite. So they are much more specific, and we need to keep on working on this. Going through to the population genetic data, I'm going to focus first on the Culex pipiens results that are the most interesting so far. So what we found is that we don't have a spatial population structure, but we have an ecotype structure. Is to say, Culex pipiens has 2 different ecotypes that have to now have been identified only using genetic methods, but their demographic history is very complex. So we have the Culex pipiens ecotype and the Culex pipiens molestus ecotype. So what we find in our area in Spain is that these ecotypes are kind of mixed here. You see that we have both in that mixture and the PCA plot, we have this cloud here where we cannot really differentiate. It's more difficult to differentiate both ecotypes and could potentially be hybrids. But we did several demographic analyses, and what we found is that this is potentially an ancestral population with a little bit of gene flow between Culex pipiens and Culex pipiens molestus and Culex pipiens pipiens. This matches a recent study that has been carried out by Lindy McWright and Yuki Haba, looking at the whole range of the Culex pipiens across the continent, when they see that the Culex pipiens and Culex pipiens molestus form are very differentiated in the north of Europe. However, in the south, closer to the origin of both of the ecotypes, the differentiation is more difficult. And this is very important because we used to differentiate these species using only 1 marker. And this tells us that we cannot accurately identify both species using only 1 marker, and we need finer data. You can barely see the map, unfortunately. But when we look at the fine scale population structure and the spatial scale, we find that Culex pipiens molestus is more abundant in urban areas, which is what we expected. And here there is a pie that it's all in orange. So this is a place where all the individuals sampled were Culex pipiens molestus, which typically can fit also in humans, as this is one of the this is like the epicenter of the West Nile virus outbreaks. So what we are seeing in the population structure actually matches what we are seeing when we are seeing the outbreaks in West Nile virus. Another very interesting thing that we went a little bit before with the fish is that we have found, and we didn't expect to find this, 3 inversions that are segregating in the population. And none of them is actually associated 100%, which is of the ecotypes. So this means that they are older than the formation of the ecotypes. One is in chromosome 1, and we have 2 big inversions in chromosome 3. And we annotated this Culex pipiens genome, thanks to Aureliano Bombarelli. And we found that one of the chromosome 3 inversions is enriched in olfactory receptors and genes involved in insecticide resistance. So there is a potential for differential evolution in there. We also did a genomic scan, and again, we can see the map, a genomic scan for several hundreds of genes that have been associated with resistance in different species of mosquitoes, including Anopheles and Aedes. And we found that 2 of them have mutations that are associated with insecticide resistance. We found insecticide resistance in the gene AC1, which is also duplicated. And this is associated with resistance to carbamates and organophosphates, which has been traditionally used in farming. We also found the distribution of the KDR gene, which is a very common mutation associated with resistance to pyrethroids and DDT in several species. And we found the most common resistance cell 1014S. And what we found here is that this was actually associated with the ecotype. So Culex molestus was Culex molestus had more of this. Culex molestus had more of this. Oh, no, the other one. I only have 2 minutes, I need to run. Interestingly, in Culex perexigus, we didn't find any population structure, no inversions, nothing. They have the smallest genome of the 4, nothing. They have a patchier distribution, we found nothing in there. So these are easy. So this shows us that target vector control in urban and outbreak-prone areas requires understanding genetic adaptations, which are linked in this case to insecticide resistance and urban adaptation. And this is incredibly important that the genome is giving us this finer picture. So we had a lot of engagement with the stakeholders. We obtained permits and co-organized workshops with the health department of the Andalusia government, the mosquito control department of the Diputación de Huelva and the Agencia de Salud Pública de Barcelona. We had meetings with them, and they are also, because they are in charge of vector control, they also gave us information on what products are being used and have been used over the years, so we can relate that with the resistance patterns that we are seeing. We developed a protocol for West Nile virus surveillance for the Spanish Health Ministry and have been in contact with vector call companies through meeting and information about the project. All of them participated in the survey. The policy relevance, we are creating a guidance document for the regional surveillance programs in Andalusia. And for the vector control companies, we would like to get some operational standards, because in this case, given what we have, it is very important to rotate the insecticides that they are using or using alternative control measures. And the scientific outputs, we have the genomic data sets that will, and open source code that will be uploaded to GitHub and Zenodo. And we have a lot of outreach activities that have been carried out with children. We've visited six schools, so we have explained this to more than 500 children, more or less, and have participated in the European Researcher Night or the College of Veterinary Medicine in Spain. We gave a talk. And the next steps are like finish the Culex perexigus and comparative genomics analysis, make all data sets available. And if we can scale up this to the whole country, we would like to create and develop a SNP panel where we can monitor resistance, if the mosquitoes are infected by the different viruses, and if they are molestus or pipiens, but we need information outside Andalusia. And this is the group, and that is doing this work. And I would really like to give a shout out to Sonia. This is part of her PhD thesis, and she has done a lot of work to get this to where it is now.
I mean, these mosquitoes are not only existing in Spain.
No.
Do you see this population structure in other Places around the world.
Yeah, so for Culex pipiens is distributed all across Europe. And what has been shown is that the difference between pipiens and mollustus does exist, but it is different in Northern Europe and Southern Europe. So in Southern Europe, we see most of this mix where animals, where the two ecotypes have not really diverged. Because they estimated that so originally, so this comes from, I don't want to make this very long, but originally it was thought that Culex mollustus evolved in the London Underground, like 100 years ago. And now they've associated that with the starting of farming in the Middle East, Egypt area. And so now they think that's why in the Southern Europe, where we also don't have the conditions to make them really different because we have winters that are milder, etc. We don't see that much of the differentiation. So we are in the southern part of that division.
No.
You can ask me later.
Thank you very much. We finish with everyone's favorite animal, the humblebee. So, oh, point is the tool. Oh, that was the wrong one.
This one. Okay. Can I start? So, good afternoon. My name is André Henriques. I'm a PhD student from the Faculty of Science, University of Lisbon, currently working within Sea. And as you can see, we are working with two widespread and generally species of wild bees, Bombus terrestris and Lasioglossum molecularum, and we are trying to understand their response, their local adaptation response to small-scale farming landscapes in Portugal. So, as we already know, pollinators are under increasing pressure due to climate change and land use change. Here in this presentation, we will focus on land use change. More specifically, we'll talk about small-scale farming landscapes. So when we think about agriculture, we also think about large-scale farming with a lot of monoculture that extends and occupies all the landscapes, with management being done by several machinery and also the application of high levels of agrochemicals. But here in Portugal, what we have is this kind of landscape, the small-scale farming landscapes, that despite the use of some level of pesticides and herbicides and also some machinery to do the management, they have some specific traits that make them kind of unique. So we have this mosaic-shaped landscape that we see here with a lot of land use heterogeneity. We have more semi-natural elements like forests and shrubs that we can see here in this land use analysis from this class here. We also have greater crop diversity and smaller field size, and we have small dispersed village that will allow species with more feeding and nesting habitats, because most of them will have small gardens and horticultures, and so they can use that as well. Since these kind of small-scale farming landscapes are very important in terms of ecology and also economically, and being Bombus and Lasioglossum also two main pollinators and two main species of wild bees, is kind of important to understand how they are coping and responding to these kind of landscapes. And so based on that, we want to integrate population genetics, landscape genomics, and also morphometical data to understand the impact of these small-scale farming landscapes in both neutral and adaptive genetic variability of these species. Because we think that this kind of mosaic-shaped landscape can be positive for pollinators, and they can act as buffer against pollinator decline. So we want to understand if that is true, and if they are adapting, which specific regions of the genome are showing some adaptive signatures. So we do not expect no major structuring or loss of genetic diversity from the populations inhabiting these small-scale farming landscapes. So we expect
moderate to high levels of genes flow between them and the ones from low agricultural landscapes, so natural areas. However, we hope to be able to detect some distinctive regions of the genome that are basically under selection due to these small-scale farming landscapes. And so in the last, we want to try to link the patterns that we see in the genetics and also the morphological data. For that, we based our analysis on the west region of Portugal, one of the most important, if not the most important, small-scale farming landscape in Portugal. So we did a land use analysis and we tried to mimic those conditions and looking for them in Portugal. So we find another 2 small-scale farming landscapes, one in the north and one in the south. And then we selected protected, conserved natural areas adjacent to this small-scale farming landscape, but separated so that we cannot find the same individuals in both of them. We collected 12 individuals in each landscape, so we distributed them evenly so that we have a clear picture of the landscapes. We preserve them, we extract genomic DNA, we send to the facility for whole genome sequencing with a mean coverage of 20 times per individual. Then we use the 2 available reference genomes for this species to do the quality control, mapping, and filtering, obtaining 2 BCF files that we used in the subsequent analysis. So now I will show you some of the results. So basically, first we looked for the genetic status of this species. We did not find any signals of population structure, neither we found significant difference in the heterozygosity or FST levels. And then we applied BiPASS. BiPASS is a population genomics software that allows us to do a contrast analysis based on a qualitative or quantitative variable. In this case, we compare the populations from small-scale farming and the ones from low agricultural areas. And we obtain our candidate outliers for this contrast analysis. And then we applied a newly developed test that is local score windows that came out in the last version of BiPASS. That instead of looking for specific SNPs, we are looking for a range of SNPs, so windows. So the software will go SNP by SNP, and in a cumulative model, they will attribute a score for each SNP, and the regions that have a score higher than a defined threshold will be considered under selection. So potentially the ones that are responding or being important for the adaptive response of this species. Here we have 2 different response for each species. So here we use the more conservative approach, and we also have a really good response with like 180 candidate windows with a lot of SNPs. And here we had to use a more relaxed approach, and we have, as you can see, fewer windows. And then we will take those outliers that we found from the contrast analysis. We will see where they match in these windows, and using the reference genomes and also the annotations files, we'll try to understand the functions of these SNPs and why they are being selected and why they could possibly be important in these kind of landscapes. Here is just for you to see that we randomize our individuals and our populations so that we could try to understand if the pattern that we were seeing was coming from the landscapes itself and now for the populations. And we don't have a response when we randomize our populations. So this is for almost bombs to verses, and we can compare it with this result, the previous result. Sorry. And with this approach, we try to show that these patterns arise from the specific characteristics of these landscapes, and because they are adapting to these small-scale farming landscapes. In terms of stakeholders and engagement, so we focus on children and high school students. Is something that I like to work on. We also work with DCNF for the permits to collect the samples, and also when entering and exiting these conserved areas. I would like to highlight some collaboration that we have with local farmers and local land owners that allowed us to use these small-scale farming landscapes and help us in some field work. And we have also established a collaboration with Med Value Institute to do workshops and on activity with some children. We participated in 2 European research nights. We developed kind of a game so that we can talk with the little ones and that they can understand what we are doing and the importance of these kind of projects as well. And why we think this is important. So basically, these results came from several climate models that predict some harsh conditions in the future. And you can see that most of them pointed to some areas that will be considered important climatic refuges. And you can see that those areas match or are nearby these small-scale farming areas. So we wanted to understand if the conditions that we see today, if the species are coping and dealing with these conditions, and if so, if these type of landscapes could be maintained in these scenarios of climatic refuge as well. So based on our results, we don't see a negative impact of these landscapes. So we hope that we could maintain this heterogeneity in land use and that this mosaic-shaped landscape could be potentially an enhancer for biodiversity, and so that we can maintain our pollinator populations healthier. In terms of impact and communication, so we have all our data public. We have 2 publications in preparation, and so this BGA project also allowed us to participate in and collaborate with other BGA projects as well. And so here we have some examples of that projects as well. And as you can see, the communication and outreach was focused on children and trying to raise some awareness in the little ones so that they could make some change in the future as well. I would like to thank my supervisor and co-supervisor, and also Alban and Miguel that work with me doing the field work and also the identification of my samples. And well, that's it. I hope you enjoyed.
For your talk, going back to how your talk was introduced, indeed, these might be my favorite animals. So I want to ask the question, do you notice diploid male problems in your population? And if not, do you think it might be because the sex-determining locus is under strong balancing selection, and so it might be one of the peaks in your genome scan?
So I do not collect males, I only focus on females so that they are diploid, so we have all the information. So I do not know how to answer that, but because we never looked for males, so I never worked, neither extract DNA or analyze some sort of data set that included males. So yeah.
You highlighted perhaps some of the landscapes that are under threat. Does this correlate with the species that are also under threat?
What?
You said some of these landscapes that are perhaps more under threat.
Not the landscapes. Well, in this one.
Yeah.
So yeah. So these are 2 different results. So this came from my land use analysis, and this is from some climate change models. And here I'm not seeing if these are threatened. What I want to highlight is if it is good to maintain these landscapes as we see today in these scenarios of climate refuge. So if they would be positive for pollinators and wild bees, if we maintain these landscapes with these characteristics, despite the use of some levels of herbicides and pesticides. And also, you know that stressors are negative for pollinators, but in these scenarios of high heterogeneity in land use, we are trying to understand if these negative stressors are somehow less present and it will have a minimal impact on the genetic status of these populations? And if so, maybe we could preserve them as we see today in these climate refugee scenarios in the future. That was more or less our idea in showing these results.
Then thank you very much, Andre, and to all the speakers on the stage. We'll start again tomorrow at 9 o'clock Portugal time.
Yes, Portugal.
Yes. Would you like to say anything, Jose?
Nope. No, not really. Just to thank everyone. And just to say that normally there will be the bus outside to take people to Vila do Conde, to your hotels. It should be here already, right? Well, we're not outside, so we don't know, but it should be there. So thank you all for this great first day, and I'm looking forward to see you all tomorrow.